A very warm welcome, both the panelists and the participants, for today's webinar organized by Sai University School of Technology. The webinar title as The Genome Frontier and How Biotechnology is Revolutionizing the Health. So, and for the next hour, you'll be exploring the future and advancements in genetic methods. So we have the speakers for today, Dr. Anil Annamneedi, Assistant Professor in Neuroscience, Sai University. And we have a very special guest and speaker for today, Dr. Sangamitra Popudi. Probably Sir will introduce ma'am. Over to you, sir.
Okay, thank you. So good morning, everyone. So let me introduce our speaker for our biotech webinar series. So Dr. Sangamitra Bopudi. She did her master's in human genetics from India, and then she moved to Germany, and then she did her second master's in integrative neuroscience. Okay, she continued in mix of neuroscience and genetics, like neurogenetics, during her PhD, and then excel in the advanced technology, the next genome sequencing. I would say she's an expert, and then she led a group, like a molecular lab in Helios Hospital in Wiesbaden, Germany. And then she moved to a industry, and currently she is working as a director, research development in Hedera. It's a startup company which are involved in cancer diagnostics. So over to Dr. Sangamitra.
Thank you, Anil, for the lovely introduction. Hello, everybody. It's very pleasing to have me in this guest lecture or webinar. As Anil introduced me very much, I don't need to go into more details of what he has told about me. So I will start with my webinar. If everybody is ready, let's explore. Can everybody see my screen? Let's make it a slideshow. Hope everybody can see my screen. Yes? Yeah, okay, cool. Okay, let's see what we have today. So the topic of the webinar is the genome frontier, how biotechnology is revolutionizing health. Why biology? Why now? I mean, you might have heard about different streams, different things like engineering, computer science, whatever, but why biology? Why does it matter? Why do we need to study it? So again, a warm welcome, but I don't want to introduce everything because Anil already introduced myself. So I'm just going to say what I'm currently doing. So currently I'm a director for NGS assay development in a company called Hedera DX, which is located in Switzerland. So what exactly we do in this company is we work with liquid biopsy. I don't know how many of you have heard about the term liquid biopsy. Liquid biopsy means you take out DNA from your bloodstream in cancer patients rather than taking the biopsy or tumor of the affected area. So directly you just take a blood draw, like how you go do a blood test, like you take the blood from the cancer patients, and then from this blood, you make the routine diagnostics of this cancer patient and then see what is affecting them and suggest possible therapies and everything. So we are basically developing diagnostic kits for cancer mainly, and then we are also we have like tons of other projects related to AI and precision medicine and real world data and for pharma support and everything. So it's all centered around cancer biology. The company is centered around cancer biology, focusing on liquid biopsy and then helping pharma companies to move forward and developing drugs. So this is the current, and I am responsible for the whole R&D in the company, and then I foresee all the projects going on, and then we try to come up with innovative stuff for therapeutic, for precision medicine. So today I'm just going to give you a glimpse just how the future looks like when we step into this world of biology, whatever we are going to talk about. So why biotechnology? As you have always heard or been hearing from your education until now, that biotechnology will be revolutionizing health, agriculture, and sustainability. It's the future. Everybody should be aware of it, and it will shape medicine, and it will bring great things in the future. Yes, everything is true. Whatever you have heard of, it is the future, because in medicine we need a lot of research, and everything. Whatever we have right now is not enough, so we need more. So biotechnology will help us in shaping this future towards medicine. So it's not just research; it's also not just biology. It's an interdisciplinary program. So biology in combination with AI, recently with artificial intelligence, and a bit of engineering and a bit of data science, all together will make this an interdisciplinary synergy where you can choose which core you want to select. So the projected biotechnology market is very huge. I mean, worldwide, it's expected to reach around 3.4 trillion US dollars by 2030, and India alone is expected to reach around 150 billion dollars. So imagine the market which is there in the industry right now. It is huge, where the pharma companies like spend like too much money developing these drugs, and in the US alone, this is like half the revenue which is potential there. So some examples, what made this market so huge is, the very famous example is the insulin production, which happened like in the 1982, which was the very first genetically modified organism, which was approved for human use directly, was this insulin producing bacteria in 1982. Imagine in 1982, people who had diabetes were using insulin from pig and other animal resources, and it was not so effective, and they also used to have a lot of side effects from using this animal product directly. So the first genetically modified approved human use insulin was produced, in which in one of the bacteria, the insulin gene was genetically modified or introduced into the bacteria. And from this, the insulin was produced, which was really, really effective for humans, for human use, and there was no many side effects. And it was approved by the authorities, which was called Humulin, which is called Humulin. And of course, recent, after 1982 or whatever, right now, they are more advanced, but the underlying technology, whatever was used in the 1980s, is still being used. And recently you have, everybody knows COVID, and these biotech vaccines, these COVID vaccines, whatever, which have been like rapidly developed within like a month, was also being used using this technology, which was from the 1980s, this genetically modified organisms like mRNA COVID vaccines and everything. And so you can see that whatever has been the base until now, we have reached to a time point where we can increase or develop in record times all these vaccines using these technologies and save millions of lives. For example, the COVID is the best example, where all the countries like rapidly made this vaccine, and then we could sustain this COVID within a very short time using the vaccination. So all these technologies are what makes biotechnology stand unique compared to the other education programs you have. And also we have also bioplastics, microbial fermentation, also recently a lot of you might have seen in a lot of social media and everything, different companies producing different kinds of technologies using microorganisms to ferment, to reduce the plastic pollution. And this is all comes from recombinant DNA technology. So next, I'm just going to talk about a few technical terms which the course or during your lifetime you might have heard about, which makes this genomics very interesting. What makes this genomics very interesting when we say every time we say genomics or AI or whatever in genomics, what does it mean actually? I don't know if any of you might have heard it or not. Human Genome Project. Human Genome Project was huge. Like what we are as humans, what we are, what's in our DNA. This is all defined in this Human Genome Project. It took 13 years for the Human Genome Project to be completed, because back in the days, in 1980s, let's say, or 70s when they started it, we didn't have this technology, we didn't have AI, we didn't have this fancy instruments or anything. So people used to really rely on very basic, simple things. And they started the Human Genome Project, which many great scientists participated in. And then step by step, year by year, they could only finish a part of our DNA. So that's why it took 13 years for them to completely finish this Human Genome Project. And it was finished in 2003. And this is like the, let's say, the brick or the foundation for the modern genomics. So by the time by 2003, we had our DNA fully analyzed and printed and everything, and all the scientists and we came to know about the DNA and about what the DNA of the human looks like, and most of the species we could also identify by this time with the along with the Human Genome Project. So what took 13 years to doing the Human Genome Project today, at this point of time, it takes 1 week. Just look at the timeline, which we have like from 2003 to 2025, 13 years reduced to 1 week. So today, a human genome can be sequenced within a week. If you give your DNA on Monday, you have your results by next Monday. So that is what now is the advanced technology we have reached with this sequencing world, which is called the next generation sequencing. So again, the time has been reduced a lot. As I said, now it can be done within a week, and within 5 working days, you'll get your reports and everything. But the cost, whatever also happened from that time point to now, also reduced like way a lot. Like in 2001, 3, whatever, it used to cost like 100 million to do this, but now it costs like $1,000 to get it done. So we have advanced so huge that the cost and the time we could like rapidly reduce and make it available to each and every person so that they can be diagnosed earlier, or they could have their reports in hand without waiting for years, or they can have their insights into it. And then what else can this genomics be used for? Genomics can be used for a lot of different things. For example, ancestry testing, like if you want to know like where your ancestry tree comes from and where are your roots are, where is your background is. We can use it for rare diagnosis, disease diagnosis, like why some rare diagnosis is happening only in a defined population or something. We can use it for pathogen surveillance, like for example, like COVID, like how when COVID came, different variants came, like they were evolving so heavily and rapidly among different countries. Then they started the pathogenic surveillance. And then obviously, not only in humans or microorganisms or related, but we can also use this genomics in agriculture very vastly. So agriculture, we can develop drought-resistant crops. We can develop crops which are resistant to pesticides, and crops which are resistant to pests, and then environment. And depending on the environment, how efficient your product will come out from this crop. So different things we can do with genomics, not just limiting to humans, but a wide variety of other fields also. And also, along with next generation sequencing, there were other technologies which were being developed during the years. These are called DNA microarrays, and also whole exome sequencing was being done. So these are like studying in depth what is happening inside the human genes and how they are expressed inside the human gene in different diseases, like for example in cancer or whatever, where we study in depth or focus very niche, for example, like choose a particular cancer or choose a particular type of disease, and then we study exactly what this gene is doing or what this is doing in particular type of cancer. The other technology which you might have heard also is called CRISPR-Cas9, which means gene editing, which is still in development or in research until now for majority of the things, but CRISPR-Cas9 has been used in therapies for sickle cell and in experimental treatments for blindness, but it is not yet approved. But CRISPR-Cas is what is the future in our hands, like where you can do gene editing and then help a lot of people to overcome their disabilities. And the next thing in genomics, or you might have heard, is AI, artificial intelligence. Artificial intelligence not only in the fields of computers or in daily life or in whatever, but also in genomics, artificial intelligence will play a major role, because with artificial intelligence, we can develop models where we can pre-identify cancer mutations, predict gene expressions used for developing different drugs and everything. So AI is going to be a huge help for the humans to develop more in genomics. Just a fun fact. So if you take out all the DNA from each of your cell, which for each human cell contains about 2 meters of DNA. So if you take out all the DNA from all your cells in your body, it's enough to stretch from the earth to the sun and back over 300 times. So this is how much DNA you have in your body, in each of your cells. So imagine how much information is within each of us. So how this genomics or biotechnology is really helping in healthcare, like really focusing on humans and medicine, and how is it being helping us in the past few years. One example is personalized medicine. What is personalized medicine? Before, when there was not much cancer research going on, and when people were not aware of genes involving in cancer or understanding the basic information or the background, what was happening in cancer, everybody used to get chemotherapy, irrespective whether they have what type of cancer, what type of stage they are in or whatever, because there was no basic research done what's happening with the cancer patients. So they just used to get chemotherapy, whatever is the basic, and then, yeah, as you have already heard or seen, chemotherapy gives a lot of side effects, like the patients become very weak, the hair starts falling off, they don't have the good immune responses, they can't really respond to normal activities. So chemotherapy was not effective in all patients. It was like plus minus like 20 to 50% was working in some patients, and some patients were not responding to chemotherapies, and then the cancer becomes worse, and then they used to be diseased. But as medicine evolved and as genomics came into play, and then with the new technologies and with all the different technologies which I have told before, like next generation sequencing and everything, for cancer research, a term came up called personalized medicine, which means the cancer patient will be diagnosed with the cancer kit, and then we will pinpoint exactly why the cancer is coming and where, which gene. And then we will try to focus only on that part of the gene or that part of the cancer patient. And then we try to treat them so that it's not a common medical routine for all the patients, but it's really, really personal that you are going until the cell and gene level and then giving the drug responsible for that so that it will affect only that point of gene, and then the patient will be very much beneficial without not much side effects, and then we will know that the drug given is specific for that, so it will be very effective for the patient. So that is called personalized medicine. And then comes different companies who developed diagnostics for this, which is called companion diagnostics, which means giving drugs related to this specific genetic biomarkers. For example, when a person has a breast cancer or ovarian cancer, we do testing called BRCA testing, which is called, they are genes called BRCA genes, BRCA1 and BRCA2 genes, which are responsible for the breast cancer. And then when a patient comes in with a breast cancer risk, we do this gene, both the genes testing, and then we see whether if the patient has these responsible genes, whether they are effective or affected or not. And if they are affected, then we give only drugs related to these two genes only, so that we don't give them all the possible drugs, whatever is available. So this genomic medicine or personalized medicine, they reduce the cost, they give better outcome for the patient. The patient is not drastically affected by giving different drugs. So all these things help for the patient. And the next therapy or the different type of therapy which is also being used right now is called the CAR cell therapy. These are like a specific used in specific context where they're used to modified immune cells, like you give a bit of genetic engineering is done there, and then you modify your cells, whatever is existing, and then you inject these cells into your body, and then these help to fight cancer types like leukemia and lymphomas. Leukemia is like the blood cancers. So this is for blood cancers, you introduce these modified cells and then help the patient in becoming resistant. Not only that, biotechnology can also help not only in cancer, obviously in a lot of different things. For example, organ transplantations, where you have to find your matched donors, and then you have to know if the donor is correctly suitable for your exact body type. And then you have to see if, even though there is 90% matching, sometimes it gets rejected. You have to do all the different scenarios to find the perfect donor. Before, donor matching was like done on just normal blood typing, but then as the genomic medicine advanced, scientists came to know that it was not only just blood type which is enough for doing the organ transplant, but much more we have to do. Like there are a lot of different things, which we have to look before doing the organ transplant, which is now helping a lot of different people to do the matching donors. And we have a worldwide registry now for organ transplants, where people can choose or have a second life with this organ transplantation. And then there are also a section of genomics called pharmacogenomics, where drug development is done in pharmacogenomics, like where a certain drug discovery or drug development, and example like antidepressant or blood pressure drugs, which kind of drugs will work best for the patient based on the patient's body type, condition, and etc. Another fun fact is, like, the first approved gene therapy was done in 2017, which was by FDA approved. FDA is from the USA, the Federal Drug Administration. And the first gene therapy was for inherited retinal disease, which means childhood blindness, like children who were like totally blind and they could not see anything at all. They had this first approved gene therapy where it is like an injection. So this is a gene therapy, and then it is due to a gene, one of the gene which is called RPE65 gene. Uh, there is a disruption in this gene is caused by the mutations in this gene, and then the children could not they lose their vision from a young age, and they will have childhood blindness. So what the scientists did, they took a virus, an adenovirus, and they modified this virus. They injected this RP65 gene into this virus, and then they made a very nice recombinant vector virus, and they injected directly this virus into the eyes of the affected patients. And when this injection is done under the eye or in the retina of this eye, the virus releases this enzyme responsible for this gene, and then it starts the gene, whatever is affected now, it produces again in the eye, and it starts the visual cycle. And then the children who could never see in their life, they could see, then start seeing little bit of light, little bit of movement. They can read like really large text. They can navigate between obstacles. It's not like they get like the perfect vision, but they get a vision which is for them is sustainable, and then which can move around in the normal world. So this was the first approved therapy, gene therapy for an inherited disease, and the first treatment to target a genetic disease. Based on this, after this, a lot of patients were given this treatment, and a lot of children were benefited from this treatment, because imagine a child who could not see at all could at least see something from this. But it's a one-time treatment with a potentially very lifelong benefits, but the treatment is still expensive till this date because of the cost involved in developing this drug. Similarly, many gene therapies are being developed right now for many different types of diseases, which we have thought like impossible back in the days, where we thought like, no, this is impossible, we can't do anything about this. Such dreadful or life impacting diseases are also being developed right now. So gene therapy will be the future for you guys, for your future generations, for your children and everything, where people will just, even for very dreadful diseases, we will have like therapy within like very soon. So what do we use this? I mean, all this research, all these different drugs and everything, wow, it's nice, but how do we really use it in real life? Does it really help the normal person who's just with us now? Yes, of course, we use it in real life. All this research is about using it in real life. So how does it get from the lab to the clinic or to the hospital, whatever we are talking about? So one thing which I told about is liquid biopsies. So liquid biopsies is the future right now for cancer patients, where you just take a blood and then you can detect what type of cancer the patient has with high accuracy. And with the newborn screening, whatever we have developed with all these technologies, newborn screening, like when the baby is born, you can identify immediately what type of rare metabolic disorders the baby has, and then you can start the treatment right away at the baby level. So that newborn screening is a huge thing, where you can find a lot of different things, early stages, and then you can avoid them. And as I said, the CRISPR-Cas, which is the gene editing system, which can be used to cure sickle cell anemia, which is still in clinical trials, but it is in development and it can be used in real world. The other major useful thing in the lab right now is prenatal diagnostics, which means when a mother when a female is pregnant, we can test the pregnancy. We can test the fetus itself when the mother is pregnant, and then see if the fetus has any abnormalities, or if the fetus is healthy, or the fetus is expected to have some abnormalities. So all this we can do with just a simple blood draw from the mother. This is called NIPT, which means non-invasive prenatal testing. You just take the blood sample from the mother, and from that blood sample, you can test the fetus DNA, and then see if the fetus is carrying any abnormalities or not. For example, like Down syndrome or some other syndromes which might affect. So prenatal genetic testings are very much widely used right now all over the world in the pregnancy. During like 3rd to 5th month of pregnancies, they do the prenatal testings so that we know that both the mother and the baby are safe, and we can move forward with the pregnancies. Another real world application is disease outbreaks. For example, COVID is one example, but then other than COVID, there are other infectious diseases outbreaks like Ebola and Zika viruses. You might have heard about them. And these disease outbreaks can be traced within days using this genome sequencing for pathogens and for microbial infectious diseases, where you can know like what caused these disease outbreaks. So some examples to give is pharmacogenetic testing. There's also another type of field or an area which I have to say, which is called pharmacogenetic testing. It helps to give the right amount of chemotherapy, like how much amount or how much dose we should give for that patient, or for the pediatric leukemic patient, which means here like children who are suffering with blood cancer. We have to give the right amount of dosage, otherwise it might be too much for them or too little for them. So pharmacogenetic testing helps to determine like what is the right dosage or what is the right amount of chemotherapy we should give it to them. And then, as I said, like other gene therapy methods for spinal muscular atrophy, where they are completely disabled, like gene therapy helps them a lot. So another fun fact is you have your microbe biome, which means whatever is surrounding you or within you or whatever you are having right now, that everything has microbiomes in there, microorganisms in there, and this microbiome contains more genetic material than what we have, and it can influence everything, whatever you do, from related to your digestion, to your mood, to what's not, everything. So these microbiomes, they play a very major important role along with us, along with our own body. Next, I want to talk about a few things about genomic data, big data. How do you can call it in different terms: genomic data, big data, AI, bioinformatics, data science, whatever you want to call. This is going to be a huge thing in the coming 5 to 10 years. Bioinformatics has been in use since the 2000s. People have been saying bioinformatics is going to be the next big thing. Bioinformatics is going to be huge in the world of biology. Even when I was studying my bachelor's, bioinformatics, like there was a huge hype saying bioinformatics is going to be the next big thing. Yes, it is the next big thing. Starting from the 2000s till now, we have developed all the base for the bioinformatics. We have developed a lot of different things where people could not imagine we could do. But from now onwards, after COVID, let's say, it had taken a huge leap, transforming bioinformatics into a different industry with AI combined, making it as one of the biggest industries right now and the biggest hot topic right now in the world of genomics. So bioinformatics, what we can do, we can do a lot of different things before we actually do something or start an experiment in the lab or start a drug in a lab. We can model all the different things, whatever we can do with this with the AI models. So you can predict a protein, how it looks like, and try to modulate a drug, try to develop a drug, and then do different structures and everything. Maybe I can give you the best example of one of the best. I mean, you might have all seen Iron Man, Avengers, where Tony Stark uses his AI model and then predicts different things, predicts different possibilities which we can be done. So that is future what we are looking into. Like with just developing an AI model, predicting different proteins, how we can develop an efficient drug or an efficient medicine which can really help the patients suffering from a disease or multiple disease maybe in the future. So AI is going to be huge, and to help train the AI or to develop this AI doesn't come out of the flower like from thin air. You have to train AI models. You have to make them understand. You have to make them learn the different things what we want to do. So to make the AI do that, of course, we need humans again to make the AI learn all these things. That is where the bioinformatician comes, or a biologist comes, or a machine learning expert comes. These persons will train the AI models in such a way that the AI on its own can predict all this. But obviously the AI also needs some help. So that's why you need bioinformaticians and all these different people or researchers who have to develop these models. And then there are also some ethical considerations regarding like, okay, we are getting all this information, we are doing all this different research, we are doing all this, but who really owns this data and who really gets to use this data? Because data privacy is a very major thing right now, today. In many countries, it's also against law that you preserve somebody's DNA in the labs, or even the data is preserved without the consent from the patients itself. So ethical, you have to take into consideration, like not random people can use all the data. It is very, very restricted and defined, and we have to make sure that this data is not breached or like reused by everybody. So this is in a different field, which is called real world data, which means people who can get access to this data and use this data and develop from this data new drugs or new whatever technologies they want to do. So data privacy is a very important issue which we all have to consider. And then obviously you might have heard about cloud platforms, different terms, but for genomics, they have like really special cloud platforms to store all this data, like Terra and DNA Nexus, and then everybody store the data there, and then you can use. There are biobanks, many biobanks, and one of the biggest biobank is the UK Biobank, which have over like data from 500,000 individuals alone. And this data is used by hundreds of people all over the world for different kinds of studies. Interesting fun fact is humans, more than humans, there is a plant or a fern, let's say, which is called Paris japonica, which has the largest genome, like 50 times more than what humans have. So imagine when we did the Human Genome Project, it took us 13 years to finish for the human. Imagine if we were to start with this fern, it would have like it would mean maybe like took like 50 years for us to finish doing this. Okay. So what lies future for biotechnology? Today, what I told, whatever it's there today, but when you join this course, when you do your biotechnology, and then in 4 years, 3 years or whatever, when you come out of the university, and then what could happen potentially, what you could be doing in biotechnology. What is tomorrow innovation happening in biotechnology? So synthetic biology, building new life forms might be scary, but not like forms means like creating new humans. It's like not creating new humans, but it's creating new microorganisms which might help the humans in different ways. Like for example, for insulin, we use different kinds of micro, as I told, they use the bacterium like that. We develop a new kind of bacterium which can help us in a different way. So this synthetic biology or building new life forms for industrial uses is going to be huge. For example, for these bioplastics, whatever the microorganisms they are using, like that, you could use. There's also microorganisms for oil spills and everything in the oceans. Like this synthetic biology is one future technology which is going to reshape the human world today. And then regenerative medicine. Instead of looking for a donor, waiting for a donor for years to match your thing, we can grow our own tissues and organs in the lab itself, and then it can be used directly. So this is where the regenerative medicine comes into play. And then comes AI. AI in designing drugs which can enter into clinical trials way before, saving a lot of time rather than starting from scratch what we do right now. And then how can biotech even help the environment? Like in climate, like we can use microorganisms, like genetically modified microorganisms to capture the carbon, to reduce the CO2 in the air, to reduce the pollution in the air, to reduce the pollutants, whatever is flying in the air. So all this can be done. And then xenotransplantation. This is a very interesting field, which I have to say, where they use genetically modified. Here I gave an example of pig organs, but in the future, of course, definitely there will be many more coming up, where you can use the organs from different animals and then you can use it for humans. It's most likely like organ transplantation, but from different species. The xenotransplantation is also a very big field which is coming up. And next thing is biowearables. As you already have seen, there are many biowearables already in the market right now. Some of them, like for example, insulin patches, where the person need not take any shots, need not take any injections right now. They just have a patch, and then they release the insulin time to time, and the person needs it. So like this, many biowearables are to be expected to come in the near world with DNA sensors, real-time monitoring of health and everything. It's not just like your tracker, it's just like your watch, which you have like a tracker where it can measure your blood pressure, heart rate, and everything. But these biowearables are be like detecting to the DNA level, to the cell level, where you can real-time health monitoring can be done. And of course, biotechnology is not only limited here on the earth. We can also have a lot of different things explored outside, like in the space, like study space biotechnology, which means studying the human genetics and solutions for long-term space missions, how are in space, how the microorganisms, if exist, different planets, when we get a sample, how the microorganisms exist there, and all the different kinds of things, whatever you can explore is a different field. So these are the innovative technologies, or you to expect what's coming in the future for your generation. So as I said, of all these fascinating and interesting things, why do you need to learn biotechnology, or why should you join in this course? Because you learn all these cool technologies, which is the gene editing, what is PCR, what is cloning, what is the sequencing technologies. You get to learn all the stuff, whatever I've shown to you or in detail, and get more insights into them, of course. And then you can also have your knowledge into bioinformatics and AI and statistics-driven science and everything. And you can think about innovation and teamwork. And then with this course, you can get some really nice industry internship and academic research opportunities where you can build your own hands-on skills and all these different technologies, or a few of the different technologies during the study course. And you can also collaborate with different projects with different people involving in different fields, for example, either in diagnostics or in agriculture or AI or bioinformatics or whatever you choose for. So you have an opportunity here to learn different things, to experience different things, and to implement also different things, to get your hands on not just, book knowledge or whatever. I say this is like really, really where you put your knowledge to the practical use. So and then you say, okay, that sounds all cool. I mean, I can join the course, and then I learn everything. And then when I finish my course, what should I do? Like, I know there's different fields and things which we can do. So what are the career opportunities which lie when you come out of this course? Like once you finish, there is called the biotech R&D, which is the standard which I will tell, like the scientist, you say, like the biologist, where you join a pharma company or a diagnostic lab or an agricultural tech company or clinical research, whatever you do R&D, like what I'm doing right now. Like I join a pharma industry company where I do R&D, and then I do all the basic research responsible for it. So that you become a scientist. And then what else is there? What if I don't want to be a scientist? Of course, there are other jobs other than being a scientist. You can become a genetic counselor. A genetic counselor is something which I told you about pregnancy tests, remember? So when a person gets this pregnancy test, and the genetic counselor is responsible to give counseling to the mother or to the parents, if an abnormality is seen in the child, like how they should handle and everything. And if a child is born with some abnormalities, then genetic counselors direct the patients or the families or help the families how they should move forward and what are the best scenarios for them. And of course, the next high growth job is a bioinformatician or data scientist or clinical scientist, AI, machine learning scientists. So all these different bioinformatics related, software related jobs come. And then comes pharma recruiters. What are pharma recruiters? You might have heard about all the big pharma companies like Biocon, Thermo Fisher, GSK, Novartis, everything. So pharma recruiters are basically biotech scientists, where they help the pharma companies to identify or find the best possible solutions for the pharma people. These are also high-end paying jobs where they should be specialized in pharma related industries. And what else do we have? We have a lot of different startups coming for personalized medicine, which I explained previously, like really pinpointing the person, what is he really affected in, and giving medicine on it. CRISPR-Cas9 related startups, AI startups. If you go to the American Silicon Valley, you see that half of the startups are based on AI biotech platforms, developing this personalized medicine, developing CRISPR things, AI models, and everything. What else do we have? In India, government bodies like CSIR, DBT, ICMR, they offer a lot of different research positions. And you can be in the research position and do a different type of R&D and research within India itself. And then there is also other exploratory fields like regulatory affairs, where the quality and management comes into play. And this is a huge industry itself, regulatory efforts. If a drug has to be approved, who approves it? You see that the Indian Council approved this drug or the FDA approved this drug, but who is that approver? There, that is the job of the regulatory affairs. Like the person who approves the drug or the person who approves something to be released into the market. So to do that is a separate thing. And then there is another opportunity which is called scientific writing, where you write journals, you write blogs, you write publishing materials, articles, and everything, which is it's kind of journalism in biotechnology, let's say. And then there is healthcare analytics, which is, I have told about the real world data, where you collect different kinds of things, and it's a different opportunity again. So, as I said, biologists, you're not just, when you think about biology, it's not just scientists or researchers. You can be entrepreneurs, you can be change makers, you can be policy makers, you can be anything, whatever you like. As I said, before it used to be like, okay, you study biology, you'll be a scientist. No, scientist is just one branch of this. You can be, there's a lot of different opportunities, as I have explained here, different things. These are just few examples. There are many more which you can do with studying biotechnology nowadays. With this, I would like to end my talk. And then, as Alan Kay always says, the best way to predict the future is to create one. Now you have the chance to create it. So join the course. You are the leaders. You are the innovators for the future. And India is just starting with this biological revolution, and you can be the first step to it. So please take interest, and if you have any questions, you can reach out to me. And my colleague now, Anil, will start with his talk, and after his session, we are open to ask for any questions or answers you have, questions you have for us. Thank you.
So excellent. Don't worry, I'm not going to take so much time. First of all, thank you, Sangamitra, for elaborative details about different fields, different aspects, and the future perspectives. So I will quickly go through a couple of slides. Might be interesting for you. Okay, to continue with what Dr. Sangamitra has explained. So I just want to, briefly introduce what we are as a neuroscientist doing, using these biotechnology or genetic tools, in understanding the health. So I just want to talk about ASD. So what is ASD? So ASD is autism spectrum disorders, which are like a group of neurodevelopmental conditions. So the clinical features or the signs or symptoms of this ASD includes, chronic problems with social communication, socialization, and stereotypic behaviors, both the motor stereotypes and verbal stereotype behavior. And also these kids, the autism kids, they have limited interests, and they are capable of, processing the information surrounding them. So not all the ASD kids have similar way of processing this information. It varies. Okay. So apart from these, clinical signs or symptoms, the ASD is often overlap with different comorbidities or other conditions such as intellectual disability, anxiety, or epilepsy. And it's a growing concern that ASD kids, they are developing epilepsy. It's not only in India, across the globe. So scientists, we trying to understand why this is happening, why there is a percentage of growth, epileptic growth in these autism kids. So autism or ASD is not a simple condition. It's actually a multifactorial or multigenetic disorder. So multifactorial in the sense, it's not only genetically inherited, but also There's a lot of environmental, factors that play a crucial role in the etiology or the progression of ASD. And on top of that, it's not a single gene driven, condition. There are more than 1,000 genes that are linked to ASD. According to this Simmons Foundation for Autism Research Initiative, SFARI, so there are 1,230 risk genes that are associated to ASD. Thanks to advancements in this genome research, like next generation sequencing or whole exome, sequencing, scientists are able to, map the genome and then identify these many diseases. So if you look into this, ASD, this SFARI database, so these are all these 1,230 genes that are associated with ASD and spans across all the chromosomes. It's not a single chromosome is a risk chromosome for ASD. You cannot say that one. Yeah? You can see that the magnanimity of these genetic component of this disease, the ASD etiology. So what are the other genetic tools that allow the scientists to understand ASD? Yeah? So though there are more than 1,000 genes that are linked to ASD, but there are commonly identified risk genes, as mentioned here. Yeah. But what we do actually, so using different genetic tools like CRISPR-Cas9 system or CRELOX system, scientists are able to edit the genome or knock out one of these genes in order to understand, in order to develop the mutant models and in order to understand what exactly happening, like, signaling behind the symptoms, what we see in these mutant mice. So there will be functional alterations or behavioral alterations. I'll quickly go through this, behavioral alterations. What are the ASD behavior? As I mentioned that social behavior is the core behavioral aspect that is altered in this ASD. So how we can model this in mouse? Yeah, this through male-female interactions. You basically place a male and female, and you always compare a control male with the ASD male and its interaction with other normal female. So here you can see the following behavior as nose-to-nose contacts, nose-to genital contacts, and so on. Usually the control mouse, they have all these kind of behaviors, yeah? Whereas these are missing in the ASD. And then there is another routinely used test is three-chamber test. So what exactly this three-chamber test? This is used to look into the sociability in the sense how this mouse interact with other conspecific, like another mouse or an objective. So usually the mouse, they prefer and go interact with the stranger one here, not the object. So the time they spent with the stranger one indicates the intact sociability. And then you can also look into social memory or social recognition, where instead of the object, you place a novel mouse, which is called stranger tool. So what is the innate behavior of these rodents is that they interact more with the novel ones. So the memory is in a way that stranger one, it's already interacted. So it remember that one, and then it's no longer interested to go and interact with the stranger one. Indeed, it go and interact with the stranger two, which is a new one. Yeah? The time they spend with the stranger two is indication for intact social memory. Another behavior is anxiety or hyperactivity, what we often observe in these ASD kids and also these mouse models. So this we can do using open field, a big chamber where you put the mouse inside, and then the time they spend in the center is indication for less anxious behavior. If they are spending more time in the corners, that is, it's a more anxious. And another test is light dark test, which is having a chamber with illuminated and then dark, compartments. The mouse is freely allowed to, choose either of these. Usually the rodents, they spend more time in the dark. So that's why when we do this test, we look into the activity in the light compartment so that we can assess the anxiety. Finally, another test is elevated plasmase, where you have this elevated cross, basically with two closed arms and the two open arms. And here we measure the time they spend in the open arms, because the anxious mouse usually go and sit in these closed arms. Okay. Now I'll just quickly show a couple of videos. So this is the example for social behavior. So you have control here. Okay. And then you have the ASD one this side. So you see the object is on their left side, where both of them in that chamber where there is object is placed in that opaque cylinder or a cage. Now you see the difference. The control one, which is a normal one, it goes to the chamber where there is a live mouse. And see that start interacting with that normal, mouse. Whereas this ASD mouse, it go back to the other chamber where the object is there, because it's not interested in the conspecific. It's not interested in, interacting with that mouse. So this is one example where you can see these sociability differences. And then I will go to the next one, the anxiety behavior or activity. Yeah. So as I mentioned, this is the time they spend in the center is the measure for less anxiety. So let's see what this control mouse does. So as soon as we put the mouse into this chamber, it start, exploring slowly corners, and then go to the center, come back, and then, well, it kind of decided to let's explore this entire chamber. So it goes freely, it moves around this open field. Yeah. When it comes to the knockout or the ASD mouse, you see how fast it moves, and then you see that how fast it goes around these corners. It's you want to go to the center, but yeah, no, I don't want to go to the center. And then I just stick to the corners. So in this way, actually, you can model this ASD behavior in, in the model organisms like mouse, rats, or people also use the zebrafish, yeah, and so on. So this is just to show you that what we are doing using these genetic or biotechnology tools in neuroscience research, understanding disease biology. Okay, with this, I just thank you, and then leave the slide with the advanced courses what we are offering as a part of BTech Biotechnology program. Well, some of them already Dr. Sangamitra mentioned about the space, and yeah, we have astrobiology course, we have protein engineering, molecular neuroscience, vaccine technology, medical biotechnology, and so on. So with this, I stop it here.